everyone, welcome to Botany Insider, and in this lecture we'll be dealing with the third topic from the genetics unit, and that is about the Mendelian principles. We have already covered the basics of genetics and also about the monohybrid and the dihybrid crosses. You need to go through these videos in order to understand about the Mendelian principles in the best way. Why? Because we'll be carrying forward the topics that we have discussed, that is about the monohybrid and the dihybrid cross applications are completely into these Mendelian principles. Hence, I'll put the links of the same in the description box so that first you go through those videos and then understand about the Mendelian principles for a better and a wholesome understanding. So now let's understand about the Mendelian principles. So talking about that, so we have Georg Mendel. So who was he? So he was the scientist of the modern genetics, and in 1856 Mendel gave a decade-long research project in order to investigate the patterns of inheritance. So it was him because of which today we are able to understand about the various inheritance laws, the various ways in which a particular gene is inherited onto the next generation. So now let's understand about the Mendelian principles. So Mendel genetics is based upon the transmission of the characters, the units, genes from the parents to the progeny, from generation to generation. Consider we have one parent, so this is the parent 1 and this is the parent 2, and then there occurs crossing between the parent 1 and parent 2. So now we have the formation of the offspring, that is the F1 generation. So how the characters from both of the parents are transmitted to the next generation, to the offspring, that is the F1 generation. How does this particular complete mechanism occurs? This is what we study in the Mendelian principles. So this is what we have studied, that the transmission of the characters, units or the genes. So in the present day we call them as genes, and when the Mendelian principles were being discovered, they were given the name of factors, and we can also call them as characters, as units in any way. So in this Mendelian genetics we are actually studying about the transmission of the genes from the parents to the progenies from generation to generation. That is, after we study about the F1 generation, then we have the F2 generation and so on and so forth. So this is what we study in the Mendelian genetics. So according to Mendel, a factor which is now called a gene is defined as the genetic factor that controls one trait or one characteristic of an organism. So according to Mendel, when a particular organism have any specific character in particular, so that particular character is controlled by one gene, which is now called a gene. Earlier it was given the name of factor. So now let's move ahead. So he used pea plant as a model organism for his investigation. That is, for the complete calculation or for the complete investigation of the laws of inheritance, he used pea plant as the model organism. So now what is a model organism first? So a model organism is the organism that makes it very easy for the researcher to investigate a particular scientific question. That is, it helps the scientist in order to understand that how the traits are inherited. So he used pea plant for the same purpose, and pea plant in scientific term is given the name of Pisum sativum. So make sure you remember about the scientific name also. But why did he use pea plant only? So there were many reasons for the same. So the first reason was that pea plant was an annual plant. That is, it was not a perennial plant for which he had to take care for a longer time. It was an annual plant, and there could occur flowering in 2 to 3 months itself. It helped him to have the working mechanism to be done in a better place. The second important reason was that the plant that he used was a bisexual plant. That is, it contained both the male and the female characters. Hence, the self-fertilization concept was very easy. There were many offspring produced in the pea plant. Hence, the study was easy because of the many numbers of offsprings produced. The plant was a diploid plant. There were many contrasting characters available, and hence, because contrasting characters were there, it was easy for him to study. The next reason was that the plant was of average height, that is, it was of small height and it could be cultivated on the small patches of land. Hence, it was easy for him to manage those plants. So these were the reasons why he chose pea plant for his investigation. And what were the characteristics that he used? So he used 7 characteristics for his experiment. The first was the form and the cotyledons of the seeds, and each of the character that he chose had the contrasting character also. That is, in case of form, the form of the seeds were either round or either wrinkled. And when we look at the cotyledons were either yellow or green in nature. Talking about the flower color, that was the third character which he chose. The flower color was either white or violet red. Talking about the pod form and the color were taken into consideration. Pod form was either full or constricted between the seeds, and the pod color was either yellow or green. Talking about the stem that he took, the size of the stem was either long or short, and the position of the inflorescence was either axial or terminal. So these were the 7 characteristics that he chose for the experiment. So now let's understand about all of the Mendel laws. So there were 3 laws of Mendel. The first was law of dominance and uniformity. The second law was law of independent assortment, and the third law was law of segregation. So now let's understand about each law in detail. The first law is law of dominance and uniformity. So what does this law state? So this law states that some alleles are dominant while some of them are recessive. An organism with at least one dominant allele will display the effect of the dominant allele. We can state it at this way also, that Mendel's law of dominance states that in a heterozygote, one trait will conceal the presence of the other trait for the same characteristic. The recessive will remain latent, but they will be transmitted to the offsprings in the same manner the way the dominant traits are transmitted. So what does this mean? Let's understand about this. So it states that the law of dominance says that whether it is a dominant or a recessive trait, the traits would be transferred to the next generation in the same way. But one thing we have to remember according to law of dominance, that when we have a heterozygote, so this heterozygote will depict the allele, or it will form the characteristic, or it will display that characteristic of which the dominant allele is present. As we can see, it is a heterozygote for the height character, and because we have learnt about the monohybrid cross, capital T represent a tall plant and small t represent a short or a dwarf plant. So because it is dominant, the tall plant or the tall character is dominant, hence this heterozygote would represent a tall plant. So this is what is actually stated in the law of dominance. So as you can see, it says that some alleles are dominant while some of them are recessive. The organism with at least one of the dominant allele will display the effect of the dominant allele. So this was about the law of dominance. As you can see that we have the phenotypes as the red cherries, yellow cherries and the red cherries. But when we look at the genotypic makeup, the red cherries is for the capital A. So from this we can make out that capital A means the dominant character, and in this particular case the dominant character is a red cherry. When we look at the second one, that is small a, so it is homozygous recessive. We have already dealt with that what is homozygous dominant, what is recessive, what is dominant in the previous videos. If you have not watched them, please do watch them for a better understanding. I won't be able to explain all of this again. That is why I have made a separate video for the basics. So please watch those videos so that you can understand these terms. So let's move forward. In the third case, we have the genotypic makeup as capital A, small a. So it is a heterozygote, but because we have one of the character being the dominant one, and we have just seen that the dominant represents the red color. Hence, this heterozygote also represented or also displayed the red cherries. So this is about the law of dominance. The second law is the law of segregation. So what does this law state? So this law states that during the gamete formation, the alleles for each of the genes segregate from each other so that the gametes carries only one allele for each of the genes. What does this mean? For this, we have to revise about the monohybrid cross. We have already dealt about this in detail in the previous video. So in monohybrid cross, the Mendel tested individually the 7 characters in his monohybrid cross. And what is a monohybrid cross? We have already discussed. The monohybrid cross is the one which possesses only a single pair of character. In this, we will be understanding or the Mendel crossed the tall and the dwarf plant. So, talking about the tall and the dwarf plant, in the parental generation, the parents were taken as the homozygous dominant for the tall and the dwarf character. This was the parent 1 and this was the parent 2. The gametes that were formed were capital T and the small t. And in the F1 generation, we had all of them as tall plant. They were all heterozygous for the combination. After the F1 generation, there occurred selfing, or Mendel did the selfing of the F1 generation, and as a result of that, in the F1 generation, we had the formation of gametes as the capital T small t from the first parent, and from the second parent we have capital T and small t. So in the F2 generation, we had capital T as the first offspring, capital T small t as second, capital T small t as the third, and small t as the fourth of the offspring. So we had 3 tall and 1 dwarf plant. So talking about the genotypic and the phenotypic ratio. So genotypic ratio was 1:2:1. We have already discussed about that how to calculate the genotypic and the phenotypic ratio. We have already discussed about that what is genotypic and what is phenotypic ratio and how to calculate them. This was the reason why I asked you to watch the previous 2 videos first, because you would be able to understand about this video only after you have dealt with the 2 videos before. So the genotypic ratio for the monohybrid cross is 1:2:1, and for the phenotypic ratio we have 3:1 as our final ratio. So the conclusion that Mendel got from the monohybrid cross was that the F1 generation showed only one trait, as we have just discussed, that the F1 generation showed all of the offspring being tall in nature. The second conclusion was that the trait which remain hidden in the F1 generation reappears in the F2 generation with the frequency of only 1/4 of the total number. So as we have just seen that in the F1 generation we did not had any of the dwarf plant in the initial case, but in the F2 generation the character, that is the dwarf character which was hidden in F1, was shown in the F2 generation, but this dwarf plant or this dwarf character was seen in only 1/4 ratio, that is out of the 4 only one of them was dwarf. So we have just discussed that we had 1, 2, 3 and 4 of the offspring, and out of the 4 offsprings we had only one of them as dwarf in nature. So this was the second conclusion, that the trait which remained hidden in the F1 generation reappears in the F2 generation with the frequency of only 1/4 of the total number. So the final conclusion was that hence the paired genes or the pairs, they separate or the other word for the same, that is they segregate from one another and they are distributed to the different sex or the gametes. So we have just seen that these genes get separated out from each other, and as a result of that they segregate from each other. That is, during the gamete formation, the alleles for each of the genes segregate from each other so that the gamete carries only one of the allele for each of the gene, as we have discussed about this in detail. So this was about the monohybrid cross and the second law. Let's talk about the next law, that is the law of independent assortment. So what does this law states? So this law states that the genes of the different traits that segregate, or the genes of the different traits can segregate independently during the formation of the gametes, or the different pair of the alleles assort independently and they segregate from each other. And for understanding this law, we have to understand about the dihybrid cross. We will just revise about the dihybrid cross in this video because I have explained about this complete cross in the previous video itself. So in the dihybrid cross, we take two of the characters in consideration, hence it is given the name of dihybrid. The two characters that were taken into consideration were, the first was the color of the seeds and the cotyledons, that is either yellow or green, in which the yellow is dominant over the green. The yellow is represented by capital Y and the green is represented by small y. The form of the ripe pods is the second character that is taken into consideration, which is either inflated or wrinkled. So let's talk about the dihybrid cross. I have explained you in detail about this in the previous video. Let's just quickly revise this. Initially we had the parents as capital Y, capital R, and they were crossed with the parent second with green and wrinkled. We had the formation of the gametes carrying capital Y, capital R and small y, small r. And after the crossing in the F1 generation, we had capital Y, capital R, small r, that is round and yellow. There occurred self-fertilization of the F1 generation, and as a result we had, after forming the Punnett square, we had the 16 offsprings produced, with 9 of them being round and yellow, 3 of them being round and green, another 3 being wrinkled and yellow, and 1 of them being wrinkled and green. We had the phenotypic ratio as 9:3:3:1. So what was the conclusion from the same? So from the dihybrid cross, there were two conclusions made. The first was that the pair of the alleles assort independently, which is similar to the monohybrid cross, and the second was that the first gene does not interfere with the second gene. So these were the two conclusions that were made out of the dihybrid cross. We have the third law as that the genes during the traits can segregate independently during the formation of gametes, and the pair of the alleles assort independently or they segregate from each other. And with this, we're done with this complete video in which we discussed about all of the 3 Mendelian principles in detail. I hope this video was helpful for you, and if you like this video, please do like, share it with your friends, and please subscribe to my channel. I'll be coming up very soon with more videos from the Unit 8 and others also. So thank you so much for watching, and I'll see you soon. Bye.